Imagine for a second that you are looking at this highly complex, just incredibly detailed satellite image of a massive city.
Right, like Google Earth, but on steroids.
Yeah, exactly.
Yeah.
You can see the shapes of the skyscrapers, the winding roads, the layout of the parks. Now imagine if, just by looking at the physical shape of those buildings from space, you could perfectly predict the exact conversations happening inside them.
Which sounds completely impossible.
It sounds like pure science fiction.
Yeah.
The exact words being spoken in the boardrooms and the living rooms just from a picture of a roof. But in the medical world, this concept is basically the holy grail of pathology.
It really is.
The idea that you could look at a simple, traditional image of a tumor tissue slide and instantly know its hidden molecular and genetic code.
Right, because traditionally those are two completely separate languages that require completely separate tests. I mean, you have the structural physical language of the tissue.
The pathologists actually see under a microscope.
All right, exactly. And then on the flip side, you have the chemical molecular language of the DNA, the RNA, the proteins, and bridging that gap has historically been, well, an absolute definition of diagnostic muddy waters.
Muddy waters is a good way to put it. And the reason we're talking about this today is because a new 2026 paper from Nature Methods has essentially provided a Rosetta Stone to cross those waters.
It's a really powerful one, yeah.
They've introduced a framework called Multi-Embed. And, our mission for this deep dive is to unpack every single mechanism, term, and detail of this unified multimodal learning framework.
We're going to get into the weeds today.
We really are.
Okay.
Let's unpack this. We are going to look at the exact math, the algorithms, and the training strategies that allow it to decode this secret language of disease so you can fully grasp how this is shifting the landscape of network medicine.
I think the broader context here is really important for people to grasp first. Previous artificial intelligence models in pathology have, they've constantly run into what we call the interpretability and generalizability dilemma.
Right, the classic AI black box problem.
Exactly. In simple terms, earlier models could often predict a handful of genetic markers, but nobody knew how they arrived at the prediction. Or conversely, they worked beautifully on a tiny curated data set in a sterile lab.
But then you put them in a real hospital.
Right. The moment you applied them to real, messy, large-scale patient cohorts, they just fell apart. So Multi-Embed is designed to solve that by changing how we map the why behind tissue architecture.
Okay, so if you've ever tried to merge two massive spreadsheets that don't share a common ID column, you know how frustrating data translation is. And we have two totally different sets of data here. On one side, we have the morphological features, which are the actual images.
The visual data?
Right. And the paper notes, they look at this on 3 scales, which I thought was interesting. Slide level, which are these massive whole slide images or WSIs.
Yeah.
Spot level, and even like microscopic single cell level.
And then on the other side, we have the multi-layer molecular profile, which is incredibly dense. I mean, we aren't just looking at one type of chemistry here.
No, it's a whole stack.
It's a massive stack. We are looking at genomics, which are the fundamental DNA mutations. We have epigenomics.
Wait, epigenomics, that's like the methylation stuff, right?
Yeah, the DNA methylation, basically the chemical tags that turn certain genes on and off. Then we have transcriptomics, which is the RNA expression showing what the cell is actively doing right now. And finally, proteomics, the actual proteins functioning in the tissue.
So we have visual data, which is gigabytes of pixels, and we have chemical data, massive spreadsheets of gene counts and protein levels. Before they can even interact, they have to be converted into a format the AI can read.
They need a common baseline.
Right. For the visual side, the paper mentions using something called UNI. If I understand correctly, UNI is a pre-trained pathology foundation model.
That's right.
Meaning it's an AI that has already looked at millions of medical images and learned the basic grammar of tissue. It acts as an extractor, turning those visual features into mathematical matrices.
Precisely. UNI translates the pixels into a dense mathematical representation of the tissue structure. And simultaneously, the molecular data is normalized and prepped into its own set of numbers.
But going back to your spreadsheet analogy, they are still speaking 2 different dialects. They exist in completely different dimensional spaces.
Which is the core problem.
Yeah.
So how do we force them into the same room?
Yeah, how do we do that?
Through what the researchers call the multi-embed space. The model uses MLP encoders. That stands for multi-layer perceptrons.
Multi-layer perceptrons, okay.
Yeah, they are a type of feed-forward neural network.
Yeah.
You can think of an MLP as a highly complex mathematical translator. It takes both of these separate sets of matrices and projects them into a shared, unified mathematical dimension.
I'm trying to picture this physically. It's like, imagine two separate funnels pouring visual data and chemical data into a shared dark room, the multi-embed space. So if I take an image tile of a few cancer cells and I run it through the MLP, it lands at a specific set of coordinates in this shared dark room. And if I take the RNA data from that same exact piece of tissue and run it through its own MLP funnel, it should ideally land at those same coordinates.
That's the goal.
But wait, that makes sense for a tiny spot of tissue. What about the slide level data? A whole slide image is huge. It's made of thousands of smaller tiles.
Tens of thousands sometimes.
Right. And a lot of that is just healthy background tissue or empty space on the glass. If you just mathematically average all those visual tiles together and dump them into the shared space, wouldn't the signal from the actual cancer cells just get washed out by all that noise?
That is a critical flaw in older models, actually. And you've hit on exactly why multi-embed uses an attention mechanism for whole slides.
An attention mechanism, how does that work?
Instead of a blind average, the attention mechanism acts like a spotlight. It mathematically assigns a weight to each individual tile based on how relevant it is to the overall molecular profile. Yeah, the model effectively learns to say, this tile full of stroma is irrelevant, assign it a low weight, but this tiny tile showing aggressive tumor growth is vital, assign it a high weight.
So it dynamically focuses on the visual areas that actually drive the genetic signature. It learns which parts of the city to zoom in on to actually hear the relevant conversation.
That's a great way to visualize it.
Okay, let's take a quick breather here. We are about 5 minutes in, so let's recap what we've covered. 1, we take images and molecules. 2, UNI reads the images while MLPs read the molecules. And 3, they're projected into a shared dimensional space using attention to focus on what matters.
Spot on.
And for you listening, here's a question to ponder. Think about your own field of work. What are 2 completely different types of data you wish you could translate into a single shared language?
It's a fun thought experiment.
It really is. All right, moving on. Getting the data into the same shared room is really just the setup. Now we get to the core of the paper, which is how they actually train the AI to find the connections between the image and the molecule without a human manually hand-holding it and labeling millions of cells.
Right. This is the self-supervised contrastive learning phase. In machine learning, you train a model using a loss function.
A loss function.
Yeah, it's essentially a mathematical way to measure how wrong the AI is at any given moment, so it can iteratively adjust its weights and correct itself.
Okay.
And the loss function in multi-embed is a composite equation made of 3 main parts: reconstruction loss, self-supervised loss, and regularizers.
Let's create a quick mnemonics for that. Remember, RSR, reconstruct, supervise, regularize. Let's take those one by one, starting with reconstruction loss.
Sure. So this handles the intramodality learning, meaning within the same language.
Yeah.
And it relies on something called the Frobenius norm.
Wait, Frobenius?
The Frobenius norm. It's a matrix norm. From a purely conceptual standpoint, if I take an image, compress it into this shared mathematical space, I need to make sure I haven't destroyed the original data in the process.
Oh, I see. It feels a lot like trying to perfectly recreate a high-resolution photo from a compressed zip file, like translating a poem to French and back to English.
Yeah.
You want to make sure it's still rhyme. You don't want to lose the original flavor.
That is a very accurate way to look at it. The Frobenius norm is a highly efficient mathematical tool used to calculate the distance or the error between 2 matrices. The MLP decoders attempt to take that compressed embedding in the shared space and unpack it back into the original image or the original gene sequence.
And the Frobenius norm checks it.
Exactly. It checks it pixel by pixel or data point by data point against the original. If the unpacked version is too different from the original, the loss goes up and the model adjusts. This guarantees the AI isn't just making up a convenient compressed shorthand that loses the biological reality of the tissue.
Okay, so we've preserved the integrity of the data. The zip file unpacks perfectly. Now we move to the S in our RSR mnemonic, the intermodality learning, teaching the visual and the chemical data to align. This is the self-supervised loss, and the paper highlights their use of triplet loss. What is that?
Triplet loss is really the engine of this contrastive learning. Imagine you pull a triplet of data points. First, you have the molecular profile of a tumor, which we designate as the anchor.
Okay, the anchor is the chemistry.
Right. Second, you have a pathology image tile from that exact same patient's tumor. This is the positive sample. Third, you grab a pathology image from a completely different patient or tissue type. This is the negative sample.
Oh, I get it. It's acting like a sorting magnet in that multidimensional space. The AI is trained to magnetically pull the anchor, the molecule, and the positive matching image closer and closer together, while actively repelling the mismatching negative image, pushing it far away.
Exactly. And it measures this push and pull using cosine similarity.
But wait, here's where I have to push back a bit. If I'm thinking about how algorithms optimize, they are notoriously lazy.
They are very lazy.
If the triplet loss is just rewarding the model for putting matching pairs close together, mathematically, what stops the model from just cheating? Couldn't it just collapse all the positive data points into one single meaningless coordinate in the center of the room just to minimize the distance to zero?
That's a great question. You've just described a phenomenon known as feature collapse or representation collapse. It is a massive problem in contrastive learning.
Ah, so it is a known issue.
Oh, absolutely. If everything collapses into a single point, the model learns nothing about the nuance of the biology. It just finds a math loophole. To prevent this, they introduce the third part of the equation, the R, the regularizers, specifically the divergence regularizer.
So it penalizes the model if things get too similar.
Exactly. The divergence regularizer stops all features from looking identical. It applies a penalty if the distinct features of different patients start looking the same in the embedding space. It forces the model to maintain the rich diversity of the data.
That makes a lot of sense.
But there's a second regularizer too, the MMD, maximum mean discrepancy.
Maximum mean discrepancy. What does that do?
Well, remember, these two modalities, pixels and gene counts, come from completely different underlying mathematical distributions. Early in the training, the cloud of visual data and the cloud of chemical data might drift so far apart in the dimensional space that the triplet loss magnets aren't strong enough to pull them together.
These just get lost in the darkroom.
Right. So the MMD regularizer works early on to gently tether the two distributions, ensuring they overlap enough for the contrastive learning to actually work. It keeps them from drifting too far apart.
I see the math there, but I want to jump back to something for a second. We talked about how whole slide images are huge bags of thousands of tiles, and they use attention weights to focus on the important ones. When you were doing this magnetic sorting, this triplet loss, how do you measure the distance between one single molecular anchor and a bag of thousands of image tiles?
That's tricky.
Because if you measure the distance to the irrelevant healthy tiles, it's going to ruin the correlation, right?
That is a fantastic nuance to bring up, and the researchers solved it using what they call a one-sided smooth chamfer similarity.
Okay, that is a mouthful. One-sided smooth chamfer similarity.
So I know, but standard chamfer similarity measures a distance between two sets of points symmetrically. But here, the relationship is asymmetrical. You have one comprehensive molecular profile and a massive set of image tiles.
So the one-sided part means
it means the model only calculates the similarity from the perspective of the most relevant image tiles against the molecular profile. It is a weakly supervised method that mathematically acknowledges that the background stroma or healthy tissue shouldn't penalize the similarity score.
oh wow. It smooths out the noise and only rewards the model when the critical cancer tiles align with the genetic anchor.
You got it.
That is incredibly clever. Here's where it gets really interesting for me. We are literally watching an AI learn the fundamental rules of human biology using geometry and spatial distances.
It's beautiful math.
It really is. Okay, we're at our 10-minute checkpoint. Let's recap the training dojo. Remember RSR1, reconstruct, keep original data intact using the Frobenius norm. 2, supervise. Magnetically pull matching pairs together with triplet loss and that one-sided chamfer similarity. And 3, regularize. Prevent the data from collapsing or drifting using divergence and MMD.
Perfect summary.
And for you listening, here is your next question. If you were the AI, what features of an image would you rely on most to guess the genetic mutation hidden inside? Just something to think about.
I'd probably look at the nucleus shape, personally.
Okay, that's a good one.
Yeah.
Okay, but theoretical math only gets you so far. I want to look at the benchmarks. Because, let's be honest, there is a graveyard of AI papers that boast beautiful math, but completely fail on real graded patient evidence.
That's very true.
Does multi-embed actually beat the existing standards?
It does, and the margins are quite significant. They tested multi-embed extensively, starting with bulk data from the TCGA dataset, the Cancer Genome Atlas.
That's a huge data set, right?
Massive. They tested across 12 different cancer types. They evaluated performance using the Pearson Correlation Coefficient, or PCC.
And what were the results?
For predicting highly variable genes, multi-embed achieved an overall average PCC of 0.37.
Now, To someone outside the field, a 0.37 correlation might sound a bit low, but predicting highly variable genes that fluctuate wildly between patients and tumor stages, is notoriously noisy. What were the previous state-of-the-art models hitting?
Older dedicated models like HE2RNA were hitting around 0.29. A more recent model, DeepPT, achieved 0.32. So a jump to 0.37 across 12 distinct cancers is a massive leap in predictive reliability.
That is a big jump.
But where the model truly separates itself is in spatial transcriptomics.
Right, spatial transcriptomics, or ST, because bulk data just tells you if a gene is expressing somewhere in the tissue. It's like taking a whole smoothie and analyzing the ingredients. ST is trying to figure out exactly where the strawberry is and where the banana is on the tissue map.
I love the smoothie analogy.
Tumors are heterogeneous, right? They have a dense core, a strutting leading edge, and immune boundary. Why is ST so hard for AI, and how did multi-embed perform?
ST is difficult because the spatial resolution is microscopic. Aligning the visual architecture of a single cell to its specific RNA output requires immense precision. They tested multi-embed against top spatial models like iStar and AlmaCLAP.
On what kind of data sets?
On highly complex, high-resolution data sets like 10x Visium, Xenium, and the single-cell level Visium HD. Because multi-embed projects everything into that unified, well-regularized shared space, it doesn't lose the spatial context. It exhibited significantly higher correlation values in every single spatial category compared to the previous leaders.
Okay, RNA expression is one thing because transcriptomics gives us a pretty decent signal to track.
Yeah.
But what about the quieter structural layers? You mentioned earlier this framework handles multi-layer profiles.
It does.
Can this thing actually look at a physical pathology image and predict DNA methylation, the epigenetic switches that turn genes on and off? Because that feels like an entirely different level of invisible.
It is, but it can predict it. It actually outperformed a dedicated methylation model called Deploy.
Wait, really?
Yeah. It can also predict protein abundance, beating out a model called WSI2-RPPA on an independent spatial proteomics dataset. It even successfully predicted specific genomic mutations, like the famous TP53 cancer mutation, outperforming models built specifically for mutation prediction like CHIEF and MMUSC.
So the AI is literally looking at pink and purple stained tissue and seeing the epigenetic switches in mutant proteins. That is wild.
It's basically superhuman vision.
But there is one mechanism in this paper that absolutely flips the diagnostic workflow on its head. Gene-to-image retrieval.
Yes, this is fascinating.
We spent this whole time talking about predicting the unseen chemistry from a physical picture. Yeah. But they ran an experiment using the EMT signature. Walk us through that.
This relies entirely on the architecture of that shared embedding space we discussed. Because the visual matrices and the chemical matrices now live in the same dimensional room, the flow of information is bidirectional.
Bidirectional, meaning it goes both ways.
Exactly. EMT stands for epithelial mesenchymal transition. It is a critical biological process where cancer cells lose their adhesion, become aggressive, and begin to metastasize or spread.
Right. Bad news.
Very bad news. And it has a very specific genetic signature. The researchers essentially use that genetic signature as a search query.
Wait, so it's like doing a reverse Google image search, but for biology.
Exactly. They input the EMT molecular profile, and the algorithm searched the shared multi-embed space to retrieve the specific physical pathology image piles that were mathematically closest to that query.
And did it work?
It did. When human pathologists reviewed those retrieved tiles, they physically showed the exact morphological features of aggressive invading cancer cells.
Wait, so we can essentially use a genetic profile as a search query to find physical evidence of cancer in a massive tissue slide. That's mind-blowing. Think about the implications of that. If you are a researcher trying to find physical evidence of a very rare genetic mutation in a data set of 10,000 massive tissue slides, you don't have to look through them manually. You just query the chemistry, and the AI hands you the physical needle in the haystack.
It completely changes how we can do research.
Okay, 15-minute checkpoint. Let's recap these benchmarks. 1, multi-embed beats the standard across 12 cancers. 2, it works at the bulk, spot, and single-cell levels. And 3, it predicts genes, proteins, mutations, and can even reverse search images using genes.
That's the highlight reel.
And for the audience, which is more powerful to you, predicting the unseen chemistry from a picture or finding the physical needle in a haystack using a chemical query.
I think the search query is the real game changer.
Which naturally brings us to section 4, real-world applications. How does this actually translate to patient care and clinical understanding? The benchmark math is great, but how does this alter prognosis and our understanding of disease progression?
Well, let's look at their spatial clustering work on TNBC, triple-negative breast cancer.
Okay, how does integrating these two languages actually help predict patient survival there?
When multi-embed aligns the visual and molecular data, it excels at identifying specific microenvironments within the tumor. In the TNBC study, it specifically hunted for tertiary lymphoid structures, or TLS.
Tertiary lymphoid structures, what are those?
They are essentially pop-up immune cell formations that the body creates right on the border of the tumor to fight it. The presence and density of these TLS formations are highly indicative of how a patient's immune system is responding.
So more TLS is generally better.
Usually, yes. By using the integrated multimodal features to map the ratio of these TLS regions, the algorithm successfully stratified patients into high-risk and low-risk survival groups.
And they backed that up with rigorous statistical metrics, right? I remember reading they evaluated it using the C-index and log rank tests to prove the survival predictions weren't just noise.
Yes, very standard, very rigorous statistical proofs.
But the paper also details how they used self-normalizing networks, or SNNs, for multimodal prognosis across different cancer cohorts. What caught my eye was how they handled the survival loss function. Because when you are looking at historical patient data, you have censored and uncensored data.
Right, which is always a headache.
Censored data, meaning patients who maybe dropped out of the study or survived past the end of the study period, so you don't actually know their final outcome. A lot of models struggle with that ambiguity.
They do, because missing data usually breaks the correlation. But the specialized survival loss function fed into the SNNs mathematically accounts for that time-to-event probability, whether the final event was recorded or not.
That's really smart.
It is, and when they applied this to external patient cohorts, like the surgeon dataset for colorectal cancer and various CPTAC datasets, it accurately predicted patient outcomes despite the messy, real-world nature of the data.
So we can accurately predict survival based on a static slide. But tumors aren't static, they evolve. And this leads to perhaps the most sci-fi element of the entire paper, spatiotemporal trajectories.
Time travel.
Yeah, time travel. Tracking the evolution of a tumor over time. If a pathology slide is just a single snapshot frozen in time, how does this model track an evolutionary trajectory?
They achieve this by combining the multimodal power of multi-embed with 3 specific downstream algorithms.
Okay, what are they?
First, they use spatial infer CNV. This algorithm identifies distinct clonal clusters of cells. It's essentially finding the different families of cells within the tissue.
Families, okay.
Second, they use graph ST to lock in the spatial geography of where those families live on the slide. Finally, they use an algorithm called Monocle 3.
Monocle 3, like a little eyepiece.
Yep. Monocle 3 analyzes the genetic differences between those families and draws the evolutionary family tree, tracing which cluster gave birth to the next.
So what does this all mean? We aren't just taking a snapshot of the tumor, we are watching a movie of its evolution. The algorithm is playing the tape backward to see where the cancer started and playing it forward to see where it's going.
Exactly. They applied this pipeline to stomach cancer and breast cancer datasets, and the results were stunning. The trajectory literally traced the biological timeline, starting at normal inflamed cells, evolving into premalignant clones, and finally arriving at the aggressive cancer clones.
Wow.
And what validated this entire map was that the trajectory perfectly matched the rising copy number variation, or CNV scores.
And CNV scores are a primary hallmark of genetic instability. As the cells get more mutated and aggressive, the CNV goes up. So the physical map drawn by the AI perfectly mirrored the biological reality of the cancer's progression. It mapped exactly how normal cells turn into cancer.
It's unprecedented visibility.
If you can pinpoint the exact moment on that trajectory where a premalignant cell tips over into full-blown cancer, that is exactly where you would want to target early interventions.
Exactly.
Okay, final sub-theme checkpoint. 1, it integrates data to find vital immune structures like TLS for survival prediction. 2, it maps the evolutionary time travel of cancer clones using those downstream algorithms. And for the audience, if we can map the exact trajectory of how normal cells turn into cancer, where along that path do you think interventions would be most successful?
I mean, the earlier the better, right?
Right, but finding that exact tipping point is the key. So let's synthesize everything we've unpacked today.
Sure. If we look at the big picture, the true breakthrough of multi-embed is that it fully decouples the morphology molecule associations.
It breaks them apart to see how they connect.
Exactly. By projecting them into a shared space and applying incredibly robust contrastive learning, It allows us to quantify exactly how much of a patient's molecular profile can be confidently predicted just by looking at the tissue architecture. It is a massive leap for network medicine and network pharmacology.
Because it opens the door to discovering entirely novel, mechanism-driven pathological processes that human eyes simply can't see.
Right. Processes that we didn't even know existed.
It is translating the secret language of the human body. But I know you always look at the data with a critical eye. What is the catch here? Is this ready to be deployed in every hospital tomorrow?
No. And we must introduce that crucial caveat. Because multi-embed relies so heavily on contrastive learning that triplet loss magnetic sorting, we talked about it, requires a massive scale of paired cross-modality data to learn effectively.
Oh, meaning you need both the image and the gene sequencing for the training data.
Exactly. It needs thousands of examples where we have both. Right now, it is an exceptionally brilliant proof of concept for understanding the interplay between phenomics, radiomics, and molecular omics. But it is not a plug-and-play foundation model just yet. It needs vastly more paired data to become a universal clinical tool.
And that's exactly how science moves forward. You build the engine, you prove it works on the test track, and then you start feeding it more fuel. Thank you for going on this incredibly dense, rewarding journey through the source material with us today. You've just unpacked the cutting edge of AI pathology.
It really is a fascinating time to be analyzing this kind of data.
I want to leave you, the listener, with a final provocative thought. We've talked entirely about human pathology today, but the underlying math projecting visual structures and invisible chemistry into a shared dimensional space is agnostic. Imagine if we applied a framework exactly like multi-embed to the environment. Say we took highly detailed satellite images of a massive rainforest and paired them with deep soil microbiome and atmospheric chemical data. Could we eventually predict the hidden genetic health of an entire ecosystem just by looking at a picture from space? Could we reverse search for areas of ecological collapse just by inputting a chemical signature? Something to mull over until next time.